Hello everyone, I hope you are doing well. So today I am here with another video on cartilage. I have already covered the basic features of cartilage and the types of cartilage, where is each type present, it means how is it distributed in the body, in the previous videos. If you have not gone through those videos, kindly go through them to easily understand this topic also, and for those videos you can go through the link that is given, that I have attached to this video. Okay, now in this video I'll be covering the histological aspect of cartilage. Along with this, I'll try to make you understand how to draw the histology diagrams very easily. Okay, so we have hyaline cartilage, fibrocartilage and elastic cartilage. These are the three types of cartilage. Hyaline cartilage, as we already discussed in my previous video, that it gives a translucent glass-like appearance. Okay, so coming to the basic structure of a cartilage, like a connective tissue, it has a matrix that is made up of proteoglycans, that is gel-like, in which are embedded fibers. So obviously we'll have a matrix, we'll have the fibers, and we will have the cells which are producing those fibers, and the cells which are producing the matrix of the cartilage, that means which is making up the cartilage. So now starting with the hyaline cartilage, how do you draw this cartilage in the histology? So the free surface of the cartilage is covered with the perichondrium. So this is the free surface of the perichondrium, the free surface of the cartilage. So we'll draw the perichondrium. The perichondrium has an outer fibrous layer and an inner cellular layer. Now you have to remember that perichondrium is made up of type 1 collagen fibers. So these are the type 1 collagen fibers you have drawn. Now obviously when there are fibers, that means they are produced by some cells. They are known as a fibroblast, and when they mature up they are called fibrocytes. So here in this layer we can just show the nuclei of those cells. Amongst the fibers, you can show the nuclei of the fibrocytes. Then, now you can label this as the outer fibrous layer. Outer fibrous layer. Then we come to the inner cellular layer. As we already discussed, the inner cellular layer is made up of the chondrogenic cells. Chondrogenic cells means when required they can transform into chondroblast, which can lay down the cartilage, and then when it gets mature enough it is changed into, those cells are changed into the chondrocytes and they get embedded in the matrix. So now we can label this as the inner cellular layer, and both of them constitute the perichondrium. This is how you make the perichondrium. So we have the hyaline cartilage, the free surface is covered with the perichondrium, which has an outer fibrous layer, inner cellular layer that is composed of chondrogenic cells. Right? Now coming to the main matrix. How is it different from others? One thing is that it is glass-like, it is translucent. So you don't have to show the individual fibers in it. You don't need to show the individual fibers in it. It is just like a glass. So you can use your pencil color and shade in it. Now which color you have to use? Mostly the matrix is eosinophilic. That means you have to draw it with a lilac pencil color. But in case of hyaline cartilage, you have to use the violet pencil color because it is slightly basophilic. It is slightly basophilic. So the very important feature, it can come as MCQ also, you should know it for the Viva also. The matrix of the hyaline cartilage is slightly basophilic. So use your violet color and slightly shade throughout the matrix. Just give it a shading. Right, uniform shading, so that it gives a translucent uniform appearance. Now there are spaces in the matrix. There are spaces in the matrix. They are known as, these spaces are called lacuna. These are the spaces where the cells are present. Always remember, in a cartilage, the chondroblasts, the chondrocytes, they are always present in a space, and you call that space as a lacuna, right? Now, how will the matrix appear around the lacuna? In the hyaline cartilage, it will be slightly darker. It will be, so shade the matrix slightly darker around the lacuna. This is a marker, so it is not giving the exact appearance as it should give, but with your pencil colors you can get that color. So you have to just shade it slightly darker. Okay, most of the students, they just draw a line. You don't have to draw a line. How is the lacuna formed? How will you appreciate that this is a lacuna? The matrix will be lighter all around, and around the lacuna it will be shading slightly darker. So by this you can appreciate, now this, we are in the lacuna, right? Now in the lacuna there are chondrocytes or chondroblasts. Now how will you arrange them? How will you draw the chondroblast? Chondroblasts have a feature that when they divide, they remain close to each other. Again, there is a very important reason why they remain close to each other. That is because the matrix is so dense that it does not allow the cells to move far apart. The matrix is so dense that it does not allow the cells to move far apart. And now, how the cells will arrange themselves? So, like a cell has divided and formed two cells, right? Now the opposing surfaces, the opposing surfaces will be, you'll draw them like this. They will be straighter, like this, flat. You can call it as flat. And suppose if they have divided, these cells are further divided, and so this is one D and the other is the mirror image of it, opposite, inverted D. And suppose if they have formed four cells, then again the surfaces which are in contact with each other will remain flat. Now in this case, which surfaces are flat? These are flat and these are also flat because they are in contact with the other two cells. So they will be arranged like this and the nuclei in between. So how will you draw it here in the lacuna? This is one, this is another. Similarly here, you can draw four cells here. The opposing surfaces will be flat. And then in between them, there are the nuclei. In between them, there are the nuclei. You can label these cells as chondroblast, and once they have moved deeper in the matrix, they become mature enough, you can call them as chondrocytes, right? Then, so what has happened now? The chondroblast and chondrocytes are placed inside a lacuna and this matrix is slightly darker stained around the lacuna. So what does it form? Now this matrix here is forming the territory. So you call it as territorial matrix. There are so many things in case of hyaline cartilage, and that is why you are frequently asked to draw the histology of maybe trachea where it is present or histological picture of hyaline cartilage. So if you draw all these features, you will really fetch good marks. So this is the territorial matrix and this is a territorial matrix, this is a territorial matrix, and the matrix in between them is known as interterritorial matrix. So I think everything is very clear. You have a perichondrium, the outer fibrous and inner cellular layer. Then you have the matrix that is slightly blue in color, basophilic, right? And it is glass. It gives a glass-like appearance, right? So you won't be making individual fibers. Then it stains slightly darker around the lacuna. Lacuna is an empty space that is in which the cells are present. In this, the cells, They divide mitotically because the matrix is so dense outside, they don't move far apart. So they give this appearance. So they are arranged like this, D, inverted D. Opposing surfaces flat. Chondroblast, when they become mature enough, they become embedded in the deeper in the matrix, and you call them as chondrocytes. And the darkly stained matrix around the lacuna, you call it as territorial matrix because it has formed a territory for the lacuna, and the matrix that is in between the two lacuna, you call it as interterritorial matrix. So that is all you have to draw in case of the hyaline cartilage, right? Now we come to the fibrocartilage. In fibrocartilage, the first layer is absent. That means there is no perichondrium. You don't have to draw perichondrium. So there will be chondroblasts, there will be chondrocytes. They will be laying down the layer of collagen fibers and will be getting embedded in them, in between them, right? The thing to remember here is, in case of fibrocartilage, you have type 1 fibers and type 2 fibers both. How is it different from hyaline cartilage? In this, perichondrium has collagen type 1, the matrix has collagen type 2. Whereas in case of fibrocartilage, it has mixed type 1 and type 2, but it doesn't have perichondrium, right? So how will you draw the fibers? So it is laying bundles of collagen fibers which will run parallel to each other. So these are the collagen fibers. The non-branching wavy bundles. And again, in between the bundles, there are spaces in which you have the chondroblast or the chondrocytes arranged in rows. So there is a bundle of fibers, again a space, the chondroblast or chondrocytes placed in lacuna, and again here they are placed in rows. The cells are smaller in this case. In fibrocartilage, the cells are smaller and placed in between the bundles in rows. Always draw nucleus in the cells. So this is how you draw the fibrocartilage. Now coming to the elastic cartilage. Obviously, as the name suggests, elastic cartilage will have lot of elastic fibers. So there are collagen fibers, but the predominance is of the elastic fibers. Elastic fibers, always remember, the collagen fibers are running in bundles, but the elastic fibers are singly placed and they are branching. Single, straight, branching. Okay. So again, you have to draw the perichondrium in the same way. So there will be a perichondrium, because the elastic cartilage does have a perichondrium over its free surface. Then the inner cellular layer of perichondrium. I think now it is very easy for you to draw. And the nuclei in the cells. The nuclei among the fibers. Right? So this makes up this layer is the perichondrium. You can label the individual layers. See, once you know what you are drawing, label each and everything. Then we have the elastic fibers. Many elastic fibers branching, not running in bundles, and then in between them there are the empty spaces where the chondroblast and the chondrocytes are present. They are larger in size in this case, and they have a nucleus. So in this case, they are mostly singly placed. So this becomes elastic cartilage. You can label them as chondroblast or the chondrocytes is your wish. Then these are the elastic fibers. So this is all about the histology of cartilage. If you have any doubts, you can ask me. Write them down in the comment section, and if you like the video, please like it and subscribe my channel. Now in the next video, we'll be moving to another topic, that is the bone. Thank you so much for your patience.